Welcome to another lecture by MedicoMedics, Genetics Chapter 5, Non-Mendelian Genetics. In this lecture, we will look at incomplete inheritance, codominance, multiple alleles, polygenic inheritance, environmental influence on genes, mitochondrial inheritance, and end with a summary. Now, in the previous lecture, we talked about Mendelian genetics, and we mentioned the laws of segregation and independent assortment. Now, when we talk about non-Mendelian genetics, we're talking about inheritance patterns that are not explained by Mendel's laws. Now, why is it important? Because it explains complex traits like height, skin color, or diseases. And it reflects how most traits are influenced by multiple factors. So it helps understanding genetic conditions that are not predicted by Mendelian ratios, including, for example, blood types, mitochondrial inheritance, and certain diseases. So some types of non-Mendelian inheritance include, as we mentioned in the previous lecture, incomplete dominance, which is a blend of traits like something, let's say you have a red and a white flower, and you get a pink flower. We also have codominance, so both alleles are equally expressed, like AB blood type. Polygenic inheritance, multiple genes controlling a trait, like skin color or height. Multiple alleles, so more than two allele variations existing, like ABO blood groups. We have sex-linked traits, so genes carried on sex chromosomes like color blindness or hemophilia. Mitochondrial inheritance, traits passed only from the mother via the mitochondria. And epistasis or epistasis, one gene affects the expression of another. So understanding these patterns is essential in genetics, medicine, and evolution, as most traits result from complex interactions rather than simple dominant recessive rules. So let's briefly look at these one by one. So we have incomplete dominance, and this is where neither allele is completely dominant, resulting in a blended phenotype. For example, crosses between red and white flowers producing pink flowers. A real-life example is snapdragons. If you ever looked at one, you see that red and white flowers could produce pink offspring. Codominance, where both alleles are fully expressed in the phenotype. Example, blood type A and B, so both A and B alleles are expressed equally. An example of this is roan cattle, where red and white hair appear together, not blended. So codominance is like seeing both red and white stripes on a flower instead of them blending into pink. Now we also have multiple alleles, where we have more than two alleles existing for a single gene. For example, in blood types, we have allele A, B, and O, and this produces a variation of different combinations. The real-life implication is that, for example, blood transfusions depend on compatible blood types. Polygenic inheritance, so traits controlled by multiple genes. For example, height. This is controlled by many genes, each contributing a small effect. Skin color, so this is determined by several genes interacting with the environment. And characteristics of this include continuous variation, so not just tall or short, but a range. So polygenic traits are like baking a cake with many different ingredients. Each one adds a little to the final result, creating therefore a range of possibilities. And then environmental influence on genes. So essentially, environment affects gene expression. For example, hydrangea color, so soil pH changes flower color. If it's acidic, it's blue. If it's alkaline, it's pink. Our height, for example, so nutrition during childhood influences final height. What are the implications? Well, nature, so genes, and nurture, the environment, work together. So your genes are like a recipe, but the environment is like the ingredients you can use. They can change the final outcome. Another one is mitochondrial inheritance. So inheritance of genes found in the mitochondria passed down from the mother. And characteristic for this is mitochondria contains its own DNA, and disorders here could affect energy production. So for example, we have a variety of mitochondrial diseases. And if you remember from our other lectures, it is in the mitochondria where our main source of energy is actually produced, ATP. An example of due to mitochondrial mutations is Leber's hereditary optic neuropathy. This is vision loss due to mitochondrial mutations. Now, genetic imprinting. So it's defined as the expression of a gene depends on whether it is inherited from the mother or the father. It's an epigenetic phenomenon where only one parental allele, so either mother or father, is expressed, while the other one is silenced. And unlike Mendelian inheritance, where both alleles are expressed equally, imprinted genes show parent-specific expression. Examples in human could be Prader-Willi syndrome, where paternal genes on chromosome 15 are either deleted or silenced, and maternal copies are naturally imprinted, so turned off, leading to developmental issues like obesity and cognitive impairment, etc. We have Angelman syndrome, where maternal genes on chromosome 15 are deleted or silenced, so paternal copies are imprinted, leading to neurological and developmental disabilities, etc. Now, why is genetic imprinting so important? Because it regulates growth and development in early life, and it plays a role in disease inheritance and susceptibility. So errors in imprinting can lead to disorders, cancers, metabolic conditions, etc. Now, anticipation is defined as a genetic disorder becoming more and more severe or appearing more early in subsequent generations. So it is like a genetic snowball. Each generation inherits a larger, more severe version of the condition. Examples of this is, for example, Huntington's disease, where symptoms worsen and appear earlier in offspring due to repeated expansion in the DNA. So repeated sequences in the DNA grow longer with each generation. And what is the concept of epigenetics? So it is defined as changes in gene expression without altering the DNA sequence. Mechanisms include DNA methylation, where there is a silencing of genes, histone modification, so changes how tightly DNA is packaged. Example, stress in early life can turn genes on or off, affecting health later. So epigenetics is like flipping a switch to turn genes on or off without changing the actual wiring. Now, what could be some applications of non-Mendelian genetics? Well, in medicine, we use it to understand complex diseases like diabetes, cancer, schizophrenia. In agriculture, use it to breed plants with desired traits. In forensics, identifying unique genetic markers for solving crimes. An example is studies on polygenic traits help predict risk for heart disease by analyzing many genes together. So in summary then, inheritance patterns include incomplete dominance, codominance, multiple alleles, and polygenic traits. Environmental factors and epigenetics modify how genes are expressed. Applications span from healthcare to agriculture and forensic and beyond. That's the end of Chapter 5. Thank you for listening. Continue now to Chapter 6.